Good morning, all. Bioinformatic approaches to investigate antimicrobial resistance in humans. I choose to focus on the pathogen Staphylococcus aureus, particularly methicillin-resistant strains, commonly known as MRSA. This presentation highlights how we can use the whole genome sequencing and machine learning models to study and predict antimicrobial resistance in S. aureus. About the abstract. Antimicrobial resistance, or AMR, is one of the most serious global threats we face today. So, Staphylococcus aureus is a common human pathogen responsible for a wide range of infections. Some of its strains, particularly like MRSA, are resistant to many antibiotics, making treatment difficult. So, in this project, I used genomic data from resistant and susceptible Staphylococcus aureus strains. I performed AMR gene detection using CARD RGI tool, compared resistance gene profiles, and used machine learning models, specifically like support vector machine SVM and random forest to predict resistant patterns. The study shows how biometrics can make resistant detection more accurate, faster, and scalable. Introduction. AMR refers to the ability of bacteria to resist the effects of antibiotics. This makes infections more dangerous and treatments less effective. One of the most problematic pathogens is Staphylococcus aureus. Normally, this bacterium can be treated with beta-lactam antibiotics like penicillin. But in the case of MRSA, the bacteria acquire genes such as MECA, which are like alter penicillin binding proteins and make the drugs useless. MRSA infections are a major issue in hospitals, leading to longer hospital stays, higher treatment costs, and sometimes death. The role of bioinformatics. Traditionally, AMR is detected using lab cultures and sensitivity testing, which can take days. Bioinformatics provides a faster, more precise method. By using whole genome sequencing, we can identify genes responsible for resistance within minutes. We can also apply machine learning, which uses mathematical models to detect patterns and train it to predict whether a new strain is resistant or not. This makes it possible to build intelligent systems that assist doctors and researchers in decision making. Objectives. Here are the main goals of this project. First, I wanted to analyze genome sequences of two Staphylococcus aureus strains, one resistant MRSA and one non-resistant. Next, I used GARD RGI to identify resistant genes from these genomes. Then I compared the gene profiles and created a dataset showing the presence or absence of specific AMR genes. Using this data set, I applied SVM and random forest machine learning models to classify the strains. And finally, I visualized the resistant gene data and ML results so they can interpret it more easy. Background. Understanding AMR. AMR happens when bacteria change in a way that reduces or eliminates the effectiveness of antibiotics. This can occur through several mechanisms such as genetic mutations, overuse or misuse of antibiotics in humans and animals, then horizontal gene transfer between bacterial species. In MRSA, the key gene responsible is MECA. This gene allows the bacterium to survive even in the presence of methicillin and related antibiotics. When resistant genes are acquired, they can spread quickly. They are making entire bacterial population resistant. Genome analysis and machine learning. The whole genome sequencing, also called WGS, gives a complete map of the DNA of a bacterial strain. This allows us to detect any non-AMR genes present. In this project, I use a CAR database which contains information on our 2,500 resistant genes. I uploaded the genome FASTA files into CARD's RGI tool, and it returned a list of resistance genes along with their drug class and mechanisms. So after building a gene matrix, I used machine learning to train models. These models, SVM and random forest, like learn from gene patterns and predict resistance in new strains based on those patterns. Okay, so methods, genome collection. For this analysis, I selected two well-known strains from NCBI genome database. The MRSA252 strain is resistant to methicillin and other beta-lactams. The NCTC A325 strain is non-resistant and often used as a reference. So I downloaded their full genome sequences in FASTA format. These files were used as input for the resistance gene analysis tool card RGR. AMR gene detection. I use the card RGI web tool to predict AMR genes from each genome. For the MRC strain, it detected genes like MEC-A, LAZ, and ERM-A. These genes are associated with resistance to beta-lactams, penicillins, and macrolides. The result also include match types like perfect or strict, which indicate the confidence of the match. So these gene profiles were saved in CSV format for the next gene comparison and data set creation. Next, I created a gene matrix where each row represents a strain and each column represents a gene. If a gene was present in a strain, I marked it as 1, and if not, it is marked as 0. So I also added a label column for each strain, either resistant or non-resistant. So this binary dataset is ideal for classification using machine learning algorithms. So it helps models find patterns based on which genes are present. Third, machine learning. So using this scikit-learn library in Python, I applied two machine learning models, SVM or support vector machine, which finds the optimal boundary between resistant and non-resistant strains, and a random forest, which creates multiple decision trees and identify which genes are most useful for classification. So after training the models on the dataset, I tested their performance using matrices like um, accuracy score and confusion metrics. So this allowed me to evaluate how well the models classified resistance and based on gene data. So current status, so far I have completed the genome collection, genome detection using CAR and the creation of gene matrix. So the both ML models, SVM and random forest are under implementation in Python and initial results look promising. So the data set is small for now, but future versions can be expanded with more strains for better accuracy. So my next steps includes the fine tuning the models and possibly applying other classifiers like KNN or logistic regression. Guys, to summarize, I downloaded genome sequences for MRSA and non-resistant strains, used CARD RGI to identify resistant genes, then built a gene matrix and trained SVM and random forest classifiers. The models showed early success in predicting resistance from gene patterns. So this approach shows how bioinformatics and AI can support faster, more accurate resistant detection, which is critical for modern medicine. References here are the tools and sources I used in my project. So CARD and NCPI for genome and AMR data, WHO and AMR fact sheets, then scikit-learn for building ML models, and research articles from PubMed for background and MRSA and resistant genes. Thank you.